Development and sex determination. This is going to be a very interesting chapter. You guys are going to have a really sensitive discussion this week. So this is kind of just the introduction to it. We all begin as a single cell, a fertilized egg called the zygote, which was made by bringing together gametes from your mom and dad, the egg and the sperm. Egg and the sperm meet. That point is called fertilization. Cells begin to develop and become a zygote. The oocyte is the official term for a girl's egg, which we learned a few chapters ago. Males and females produce gametes in their gonads. Their gonads are their science name for the sex organs. Testes in males produce spermatozoa, male sex hormones called androgens. Ovaries of females produce oocytes, female sex hormones called estrogens. Early in gamete formation, meiosis, that nice little process, cuts our chromosome number in half, so we only pass half of our chromosomes onto our children. So our chromosome number doesn't duplicate, doesn't double every single time we produce offspring, because that would be bad. And then fertilization unites a sperm and an oocyte, forms a zygote, from which the new individual develops and restores the chromosome number to 46. And the cycle goes on. The first few slides are about the sex organs of males and females. You all know which organs you have. You're going to learn which organs the opposite sex of you has. There's a whole table. It's a table in your book. The component: testes, epididymis, vas deferens, accessory glands, urethra, penis, scrotum. They all have different functions. And then there's graphs too, figures too. These are always fun, but I don't need to necessarily go through these. You guys are going to have, this is what it looks like from the front, the male. And then you have the cross-section of the seminiferous tubule where all the good stuff happens. This is where sperm actually forms. Mitosis, meiosis, and then incomplete cytokinesis produce haploid cells with half the number of chromosomes that differentiate into mature sperm. And then part C is the structure of a mature human sperm. The head, the midpiece has a lot of mitochondria. Mitochondria is for energy because those little guys need to swim. And then the tail for swimming too. And then there's a video there that you guys can watch on sperm production. It's the same video that we had, I think, 2 chapters ago from chapter 5, but we're reviewing the same process again, how sperm is made. And then the female reproductive system: ovaries, oviducts, uterus, vagina, all of their functions, all there. You guys all have this diagram. You guys all have this. Oocyte development. There's a lot to developing eggs. And every single month, this process happens where ovulation, the mature follicle ruptures, releasing the oocyte and the first polar body. Right before girls get their period, this is what happens. And then the human oocyte is the largest cell produced in the body, large enough to be seen with the naked eye. And again, that's actually the same video that I posted a couple chapters ago about oogenesis and embryonic development. You guys can review all the organs by yourselves. I don't need to lecture you about it. It's always awkward. You have pictures in your practice questions, and on your exam this week, you're gonna have diagrams. You're gonna have to label them so you guys can practice doing it then. Another table, 7.3, a comparison of the duration of meiosis in males and females. You can see spermatogenesis for males lasts about 48 days to go from beginning to end. And females begin during embryogenesis, which is when we're in our mother's womb. Our female reproductive cells, the oocytes, the eggs begin there. You're born with all the eggs you will ever have, females are. But the entire process to get to mature eggs takes 12 to 50 years. You will ovulate from about 12, that's about when puberty begins, when girls get their first period. Some are a little earlier, some are a little later. And then 50 is about when menopause happens. Some women are earlier, some women are later. All right, fertilization is the fusion of an oocyte and a sperm, formation of a zygote, formation of a blastocyst by cell division. So depending on how many cell divisions after an egg and a sperm come together, there's different names. You go from a zygote to a blastocyst, eventually you're an embryo. It occurs during transport to the uterus. So you have these tubes called fallopian tubes. The egg travels down these tubes, and that's where fertilization happens. And then a trophoblast will form the structures that support the fetus during pregnancy. You can see in the picture, the inner cell mass is what will eventually become the embryo and the baby. And the outer layer of cells will become support systems, support structures. Implantation, the blastocyst embeds into the endometrial lining of the uterus. Women do not carry babies in their stomachs. They carry it in an organ called the uterus, which is right below your stomach, right below women's stomachs. And then there's 3 trimesters to pregnancy. During the first trimester, all the organs form. Second trimester, the organs mature and develop further. And the third trimester is for rapid growth. The baby grows a lot in the last 3 months. So there's an overview of it, I think. Yep. So if you exit out, if you guys aren't full screen on yours, on the bottom, the next few slides, there's going to be a lot of information on the bottom. So this is a figure in your book. Fertilization, sperms surround the secondary oocyte, secrete enzymes that dissolve the outer barriers surrounding the oocyte. Only one sperm enters the egg. And at the moment that that sperm enters the egg, it actually produces a chemical reaction that'll repel the rest of the sperm to say, I'm already taken. Everybody else get away from me. Penetration stimulates the oocyte to begin meiosis too. Remember, we talked about women's eggs, females' eggs. They stop during embryonic development. They only go through meiosis 1. They don't go through meiosis 2 unless they're fertilized like this. And the sperm tail degenerates and its nucleus enlarges and fuses with the oocyte nucleus after meiosis 2. And then after fertilization, a zygote has formed. And then figure 7.6, development from fertilization through implantation. So you can see it goes through a lot of different cell divisions. You go from 1 to 2 to 4 to 8 to 16, all the way to trillions, which is us. And a blastocyst forms. Its inner cell mass gives rise to a disc-shaped early embryo. As the blastocyst implants into the uterus, cords of chorionic cells start to form. When implantation is complete, the blastocyst is buried in the endometrium. And then this is a really cool video of fertilization. I don't know why it won't play for me. There we go. It's only 48 seconds, but you can see the actual zygote splitting into two, and then into four, and then into eight, and that just keeps happening until the actual embryo forms. And it's really cool that science has cameras that are this small to be able to catch this moment of cell division. You can't even see how many cells there are anymore, but look, you got the blastocyst forming. But that's a cool little video. Back to small. All right. All right, more information, days 10 to 11. The amniotic cavity starts forming and the start of the embryonic disc here. You got blood-filled spaces all around. The amniotic cavity is going to be the amniotic fluid that the base surrounds the baby eventually. The yolk sac becomes the placenta with all the nutrients. There's week 4. As eventually the embryo and the yolk sac. And this is what the baby looks like up close. Or the embryo, it's not quite a baby yet. All the organs are just starting to form. Pharyngeal arches will eventually become lungs, developing heart. The upper limb bud will eventually become arms. The neural tube eventually becomes your spinal cord and your vertebrae. Lower limb bud will eventually become legs, and then tail. When we're at embryos, we actually have a tail. As we grow out of it, but. And then fetal development, as at weeks 5 and 6, the head grows, exceeds the growth of other regions. That's why all you can see a lot of the times is the head because it's so big. You got your retina forming, which is your eye, future ear, and then your differentiation into hand plates and then fingers, umbilical cord formation, and then your little foot plate. Week 8, final week of embryonic period before it's considered a fetus. Embryo looks human compared to the other vertebrate embryos. Upper and lower limbs, you can actually see little arms and fingers and legs and toes forming. And then the primordial or early tissues of all internal and external structures have now developed. So now they're going to rapidly mature. Tail has become stubby. Just a little stub, it's not an actual tail anymore. And there's different growth measurements at week 16, 6.4 inches, which is a little over half a ruler. If you think of how long a ruler is, it's half of that. Week 29, almost 11 inches, so almost a full ruler long. And then week 38, full term, 20 inches. That's just the average. Some babies are bigger, some babies are smaller. And then birth. Birth is hormonally induced. When the baby is ready to be born, the mother gets a flood of oxytocin, which is a hormone in your blood. Everybody has oxytocin. We just have extra when you're about to have a baby. Stimulates uterine contraction. So as I said, the baby is in the uterus. It is not in the stomach. At no time is the baby ever in the mother's stomach. It is always in the uterus. And the uterus contracts. All the girls know what it's like to have cramps when you're on your period or right before your period. They're really, really bad contractions, just like that, and it pushes the baby out. I know, it's a lovely process. There's lots of blood. The cervix gets wider, expels the fetus and the placenta. So 2 things actually come out when you have a baby, the baby and then the placenta comes out right after. Teratogens. Teratogens are always a risk to the developing fetus. These are chemicals that can cause birth defects, abnormalities. X-rays is a teratogen. If you've ever had an X-ray done, you know they always ask you, well, obviously you don't know, but they always ask females if there's a chance they could be pregnant because X-rays can cause birth defects. And then whoever the X-ray technician is, they put on this apron that's made of an impenetrable, like kind of a metal, but it's for protection for internal organs and just in case somebody's pregnant and they don't know it. Viruses can cause birth defects. Certain medications can cause birth defects. I know there's a certain acne medication. If doctors prescribe the acne medication for girls, they also have to prescribe birth control if you're a certain age because it causes really bad birth defects. So girls can't be on the acne medication if they're not on birth control. And then alcohol. Alcohol is a huge one. It's the leading cause of birth defects. There's something called fetal alcohol syndrome if a mother drinks too much while she's pregnant, especially in the early stages. There's different teratogens and then the sensitivity to them. If a mother is exposed during the first few weeks, up to the first 16 weeks, they'll have problems in the central nervous system. The first 6 weeks, the fetus might have heart problems. That's how this chart works. If they're exposed to a teratogen in the first 8 weeks, they might have problems with their eyes. Fetal alcohol syndrome is the leading preventable cause of birth defects. Meaning it's completely preventable. If the mother stops drinking, it won't happen. Alcohol has teratogenic effects throughout pregnancy, can cause miscarriage, growth retardation, facial abnormalities, intellectual and learning disabilities. And then sex determination. This should be review. We went over this. Females are XX. There are individuals with only one X. They are still female. They have Turner syndrome. Males are typically XY. Individuals with a Y are almost always male. We'll talk about the exceptions here. All right, sex determination. However, having an XX or XY chromosome set does not always mean someone develops as a female or a male. The outcome depends on a lot of things: the distribution and expression of genes on the X and Y chromosomes, interactions between genes on the X and Y chromosomes, along with many different environmental factors. So just because you have an XX or an XY, they interact with different ways with your other cells, your other chromosomes, each other, the environment, to produce many different phenotypic effects. Your phenotype is what you can see on the outside. Sex determination. In other mammals, in other animals, sometimes sex is determined by temperature. This isn't necessarily true for us, but other things are true for us. This is just an example. So alligators, if their eggs are fertilized at higher temperatures, there's a higher percentage of males. There's turtles, a lot of turtles that are the opposite. If the eggs are fertilized at lower temperatures, there's a higher percentage of males, or there's a lower percentage of males, I'm sorry. So alligators, warm temperatures means more males. Turtles, warm temperatures means more females. Those are some environmental factors that can affect certain animals. Human sex ratio. So because males produce approximately equal numbers of X and Y bearing sperm, it seems obvious that males and females should be produced in equal proportions. So the ratio would be 1 to 1, one male for one female. And this is not the case. Estimates include that more males than females are conceived. So at birth, the secondary sex ratio is about 1.05, which means there's 105 males for every 100 females that are born. This is at birth. This is when babies are born. As the generation ages, the sex ratio changes. More males than females die in childhood. And at age 20, the tertiary sex ratio is close to 1 to 1. So it's close to even. So before age 20, males tend to be a little more reckless than females do. They take more risks. They drive cars faster. They get in more car accidents. This is why if you get insurance, if you start to drive and you get insurance, car insurance for boys is actually higher than for girls. So beyond age 20, females actually outnumber males. So if we're just talking about the number of babies conceived, there's more males. And after the age of 20, there's more females. Genetic and environmental factors are responsible for the higher death rate among males. The expression of something called a deleterious X-linked recessive genes is one cause of male death in both prenatal and postnatal stages of life. So those X-linked diseases where males only have the one X chromosome, they don't have a second one to mask the recessive alleles. Some of those X-linked diseases are fatal. They can cause death. And then between the ages of 15 and 35, accidents are the leading cause of death in males, like I just said. Boys and men tend to take more risks, more physical risks than girls do. Defining sex in stages. Chromosomes, gonads, and hormones. Each of those contribute to sex and gender. So genotypic sex is established at fertilization. Genotypic is your genes. We use the letters XX, XY, or some variation. And there's phenotypic sex, what you see on the outside. That's determined by gene action, how they react with one another, interactions within the embryo, interactions with other embryos in the uterus. So if they're twins or triplets, interactions with the maternal environment. Is the mother taking care of the embryo properly while it's inside her? Stages of sexual differentiation. Chromosomal sex of an individual may differ from their phenotypic sex. The result can be a phenotype opposite to the chromosomal sex, a phenotype intermediate in between those of the two sexes, or a phenotype with characteristics and genitals of both sexes. The sex of an individual can be defined at several levels. It's not just what you see on the outside as boy or girl. So chromosomal sex, like we said, established at fertilization. Gonadal sex, the gonads are the testes and the ovaries, the sex organs. Gonadal sex is the development of ovaries or testes from undifferentiated gonads, which we'll talk about a little bit more in a minute. It's initiated by gene expression pathways to form either male or female gonads in the normal typical pathway. That normal typical pathway isn't always taken, or something could affect it. Something could alter it. And then phenotypic sex, what you see on the outside, development of external reproductive structures. Although the chromosomal sex is established at fertilization, the internal and external sex organs do not begin to develop until the 7th or 8th week of pregnancy. That's why they don't even offer to determine the gender of a baby until a certain week, till like 16 or 20 weeks. Because before week 8, you can't even tell because nothing's developed. Until this time, the external genitals of the embryo are neither male or female. Inside the body, non-specific gonads and duct systems are present. SRY is a certain gene present in males. So the presence and expression of one or more genes on the Y chromosome causes the nonspecific gonad to develop as a testes. So a gene called SRY, the sex determining region of the Y, located on the short arm of the Y chromosome, activates the expression of other genes, plays a major role in testes development, produces the excess testosterone that males have. So normally, if you have that gene, it will induce the nonspecific gonads to start producing male sex organs and hormones. The testes secrete 2 hormones. Testosterone is the main one. Stimulates something called the Wolffian ducts to develop into the male duct system, the seminiferous tubules, all of that. Along with dihydrotestosterone, abbreviated DHT. A lot of hormones are always abbreviated because they're really long. Not everybody likes to say them all the time. Stimulates the development of external male structures. And the anti-Mullerian hormone, or AMH, stimulates the regression of Mullerian ducts. The ducts need to go away to be able to function properly as a male. So you can see there's a lot of steps that need to happen for, right now we're talking about males, for males to develop normally. You got the gene, you got to have the presence of the gene, induces the hormones induces organ development. So there's a lot of steps where something could happen. In the absence of a Y chromosome, so the girls, XX fetus, the nonspecific gonads that are there up until week 8 develop into ovaries. The lack of testosterone and AMH causes the development of the Müllerian into female structures and regression of the Wolffian duct. The Wolffian duct leads to the male structures. Girls don't need that. They need the other one. The lack of DHT causes development of external female structures. Again, there's a lot of steps to develop into a quote unquote normal female. So here's just an overview of the differentiation. Appearance of an uncommitted duct system of an embryo at 7 weeks. These embryos could go either way depending on what hormones they have or what hormones they lack. They have their Y chromosome and an active SRY gene. That gene induces testes and the male sex organs. If they don't, if it's absent, it's not there, then ovaries are produced and all the female sex organs. There's the different steps of sexual differentiation. There's a lot of steps that need to go just the right way to become either male or female. You got the chromosomal sex, the gonadal sex, and the phenotypic sex. But mutations can result in gonadal and or phenotypic sex not matching the chromosomal sex. And it occurs in 1 in 2,000 births and may have several causes. So 1 in 2,000, if you think of the millions of babies that are born each year, is actually very common. So there's a lot of steps that need to go just right. And in 1 in 2,000 births and babies, something is off in one of those steps. So a few causes: chromosomal events that exchange segments of the X and Y chromosome. There's mutations that affect the ability of cells to respond to the products of Y chromosome genes. So the products would be the testosterone. You don't respond to the testosterone or the AMH, the anti-Müllerian hormone, just right. There's an auction of some autosomal genes that control events on the X and or Y chromosome. So there's a lot of reasons that people might not develop into full females or full males. Well, if something goes wrong with any of those steps, then you end up with an intersex person. Examples: complete androgen insensitivity. It's a mutation in the androgen receptor gene. The XY fetus cannot respond to androgens, therefore they develop as phenotypic females. They don't respond to the hormones as the normal pathway would indicate. So there can be fetuses with XY chromosomes that develop phenotypically as females. Gonadal intersexuality. Inefficient production of testosterone and DHT. They don't produce enough. They don't produce the standard amount. All of our hormones inside of our bodies have a certain amount to be normal. And when the hormones are off, a lot of things could happen. Bipolar is a hormonal disorder. One hormone is off, not enough is produced, or maybe too much is produced. There's a lot of mental illnesses that are just their hormone imbalances. Gonadal intersexuality is a hormone imbalance. They don't produce enough testosterone and DHT. So they're chromosomally male, but phenotypically female again. And at puberty, testosterone stimulates male sexual development. So they're born phenotypically female, but at puberty, the testosterone kicks in and it switches over. Dosage compensation is another situation, a process that equalizes the amount of X chromosome gene products in both sexes. Females don't have double the information because they have two X's. There's an actual hypothesis that only one X chromosome is active in each cell in female mammals. And X inactivation is very random, just like meiosis and mitosis and all the other processes in our body. The other inactivated X chromosome is called a bar body and can come from either parent. So just because girls have two X's doesn't mean they have double everything that the X produces. Mammalian females can be mosaics of X chromosome gene expression. So there are some mammals where they get a little bit from each X chromosome. One isn't totally inactive. Some cells express alleles from the mother's X chromosome. Some cells express alleles from the father's X chromosome. Calico cats are an example. Calico cats are the cats where you see a bunch of different colors. Normally it's orange, black, white, maybe some gray. But they're a mosaic of their parental genes. Some colors come from mom, some colors come from dad. Inactivation occurs early in development. It is permanent, except for germ cells, which is the sperm and eggs. All descendants of a particular cell will have the same X chromosome inactive. So whatever X chromosome I have that is active in my body and my cells is the X chromosome I pass to my daughter. Because genes on only one X chromosome are expressed in females, this equalizes the amounts of products from X-linked genes in males and females. There are a lot of genes that are just on the X chromosome. Females do not produce double. It just means only one X chromosome is active. There can be inactivation of an X chromosome. It begins and is regulated from a region on the X chromosome called the X inactivation center, or XIC. This is how the inactivation happens. One of the first steps is expression of the XIST gene located on that chromosome. So if you remember, you're looking at chromosome, there's a bunch of different bands. Each band controls a different thing in your body. So the XIST gene would just be a band on the X chromosome located on a larger region called the X inactivation center. When it's expressed, the X chromosome becomes coated with XIST RNA. And this causes almost all genes on the coded chromosome to become inactivated and form the Barr body. Barr was probably the scientist that discovered it, and that's why it's named after him. Random X chromosome inactivation. Since X chromosome inactivation is random, it's possible for all or almost all of the paternal or maternal chromosomes to be inactivated. Like I said, we only use one or the other. Therefore, there can be identical twins with identical genotypes, but they can have different phenotypes for X-linked traits. One twin could express mom's X-linked X, and the other twin could express dad's X. These would, of course, be female twins that both have two X's. So it would cause, even though they're identical, that would cause them to be just a little bit different physically, and only in their X-linked traits. There are also sex-influenced traits. There are traits expressed in both males and females, but expressed differently in each sex. They can be dominant in one sex and recessive in another. They're normally controlled by autosomal genes, so not the XY. Everything else, all the other chromosomes. And they're influenced by the effects of hormones on gene expression. Like I said, each of your hormones in your body have to be expressed in a certain amount. You have too many hormones, it could be one problem. You have too little hormones, it could be a completely different problem. Male pattern baldness is one of those. It's expressed differently in women than men. And it obviously happens more often in men. Hormones interact with that gene, the baldness gene, in different ways for males and females. And it's expressed more dominant in males and then recessive in females. So females can pass it on easier to their sons because they only have half the information. They only have one X. And then sex-limited traits. Genes are inherited by both sexes, but only expressed in one sex. Examples, genes involved in breast development in females, and then facial hair in males. Males have the gene for breast development, and females have the gene for facial hair, but they're generally not expressed. They're only mainly expressed in the one sex. All right, your assignments this week. There's actually 2 ed puzzles. They're both really short. I couldn't find one with fertilization and embryonic development in one, so I just did 2 of them. One's 5 minutes and the other's 9 minutes, I think. And then you have a discussion. The discussion is pretty sensitive. It's about gender and sports. There's a lot of discussion going on right now, especially at the Olympic level. If a person wants to compete in the Olympics, of transgendered or intersex, mainly males competing as females because males are typically have more muscle mass and are stronger. Can they compete as females? But there's 2 different situations that you're comparing, and they have to do with the stuff, like I said, the different pathways, the different testosterone levels, the different hormones. But that's that. And you are all considered college students. And this I put in your discussion to be respectful and be sensitive of one another's opinions. I expect you to have the maturity level to be able to do that. And then the graph is actually from an article that's in there, but it explains the scientific difference of gender expression, the gender spectrum. And it's not just male and female and no one can exist between, like on the bottom. Gender is what you call bimodal and has a spectrum. Like I said, there's chromosomal sex, gonadal sex, phenotypic sex. There's all kinds of differences depending on the chromosomes and hormones and how they interact with each other. All right. That is all I got for you guys today. If you have not, so if you got here late, make sure you take the survey. I put it in the chat one more time. If you took the survey, then you're good. Anybody have any questions? Give me a yes or no in the chat box. No, Jasmine, Jordan, Julian, okay, Shay, Alexandra, all right. So for now, you guys are good. Finish your notes, rewatch anything you need an explanation for, and let me know if you guys have any questions. I'll see you guys Wednesday.
Bye, Ms. Reese. Take care.
Bye, Maria. Take care. Have a good week.
Hi, this is Louise.
Bye, Shay. Have a good week.
You too.
Bye, Jasmine. Bye, Nicholas. Adrian, Kevin, Chioma, you guys have questions?